So this is an animation about DNA replication, which occurs during the S phase of the interphase. So remember that the interphase is when a cell is not dividing, and the interphase is divided into three phases, G1, S, and G2. So DNA replication occurs during S phase. So on top of your screen here, you can see what happened at the chromosome level. So this is one single chromosome, okay? And at the bottom of your screen, you can see what happens at DNA level. So remember that a chromosome, a single chromosome, is made of two strands of DNA. So this is exactly the same as this, but it's not the same scale. It's not the same level. Here it's a chromosome, here it's DNA level. And if you look carefully on your screen, you can see the two strands of DNA in the single chromosome. So, replication. The first step of replication is when an enzyme called helicase, you can see the enzyme helicase here on your screen, will bind to the origin of replication and will separate the two strands. So look carefully on your screen. This is what you are going to see. So here the helicase binds to the DNA, to the chromosome, and separates the two strands of DNA. So it creates what we call the replication fork, la fourchette de réplication en français. So this is what you can see here. You can see a replication fork here and another on the other side. And the two replication forks together form what we call the replication bubble. In French, l'œil de réplication. So if you need another picture, another diagram to understand this is one. So here you have the two strands of DNA. They are separated, so it creates the replication bubble, and a replication bubble contains two replication forks, one here and another one here. So back to the animation. So when the helicase separates, this is the first step of replication. It separates the two strands of DNA. And then you have another enzyme that will bind to the old strands of DNA. This enzyme is called DNA polymerase. So this is what you see in blue here. And you have another DNA polymerase that binds to the other strand of DNA. So this enzyme, DNA polymerase, will create two new strands of DNA using the old strands of DNA as a template. A template, c'est un modèle en français. Okay, so here you have the old strand in yellow, and here you have the other old strand of DNA, and the DNA polymerase will create two new strands of DNA using the old strands and what you can see here on your screen, what we call the free nucleotides. So they are separated nucleotides that are in the nucleus of the cell, and the enzyme DNA polymerase will associate the free nucleotides together using the complementary rules. So in front of a T, the enzyme will put an A. In front of a G, the enzyme DNA polymerase will put a C, etc. So it creates two new strands of DNA using the complementary rules, A with T and C with G. So this is what you can see on your screen. So I'll let you understand what happens. And if you look carefully of what happens at the chromosome level, you will see that the replication bubble will grow. Okay, first of all, it's a small bubble, but the replication occurs and the bubble will grow up to the different bubbles join together, and this is the end of replication. So here you can see on your screen that at the chromosome level, this is what happens when the different origin of replication joins together. And here you have what we call a double chromosome made of 2 molecules of DNA. So if you look carefully here, this is one chromosome, one double chromosomes, and you have 4 strands of DNA, 2 molecules of DNA. So this is how replication occurs.